Major depressive disorder can be characterized by many symptoms, including hopelessness, the inability to feel pleasure or anhedonia, social withdrawal, depressed mood, and decreased motivation. The ventral tegmental area is located in the midbrain and is part of the mesolimbic dopamine system, which is related to reward, hedonic, and motivated behaviors. Dopamine neurons in the VTA have been hypothesized to play an important part in depression, as they have a crucial role in mediating stress responses, including determining susceptibility versus resilience to stress-induced depression phenotypes. We just do not have a great understanding of why people become depressed, nor do we have a solid understanding of the underlying mechanisms and circuitry for depression, as it is a very complex illness. The combination of behavioral and optogenetic techniques can provide researchers with rich data to better understand and eventually find treatments for this disease. Rodents are often used as surrogates for human symptoms to study the neurological basis of mental illnesses such as depression. Chronic stress protocols are commonly used to induce depression-like states, and then depression symptoms are then assessed using specific tasks. The sucrose preference task is used to measure anhedonia, or the inability to feel pleasure, which is a common symptom or aspect of depression in humans. In this task, the mouse's cage is set up with two solutions, both water and sucrose. The sucrose preference is then recorded either by measuring the lit contacts of the sucrose bottle, the amount of sucrose consumed, or the difference between water and sucrose consumed by the mouse. A normal mouse that is not depressed would prefer the sucrose water over the regular water, while mice with induced depression tend to show a decrease in the preference of sucrose, which is indicative of anhedonia. The tail suspension task is another assessment that is commonly used to measure reduced motivation or behavioral despair, which is another common aspect of depression in humans that can be adequately measured in rodents. In this task, a mouse's tail is placed between two strips of tape and is suspended on a horizontal bar well away from the ground. The mouse is suspended in a way so that it is not able to make contact with any objects, including the bar and the ground. The amount of time spent struggling is then recorded. A mouse without a depressive-like phenotype would spend much more time struggling in this paradigm as compared to a mouse that shows depressive symptoms. This test measured a decrease in motivation and an increase in behavioral despair, as a mouse that is more depressed is more likely to have less motivation to get out of this test, therefore decreasing the time spent struggling. Finally, the social interaction task is used to measure social avoidance and withdrawal in rodents. In this task, a novel mouse is placed in one area of the cage, while the experimental mouse is placed in another area and is allowed to move around freely. The target mouse is free to either interact with the other mouse or stay away from it. This interaction data is recorded and coded, usually using grids like shown in this video example. While the behavioral aspects of measuring depression are very important and useful, the technique of optogenetics has greatly expanded the field of neuroscience and the understanding of the mechanisms of mental illness such as depression. A combination of optogenetic manipulations and behavioral tests have provided researchers with rich data on the relation between dopaminergic neurons in the ventral tegmental area and depressive-like symptoms and phenotypes. Dopamine neurons involved in reward and motivation have been hypothesized to be relevant in major depressive disorder. In the past, it has not been possible to test this hypothesis directly, as existing interventions are unable to specifically target dopamine neurons. Optogenetic manipulations give researchers the direct and causal information of it and how dopamine neurons contribute to depression and depression-like phenotypes. Optogenetics is a new technique which allows researchers to control the behavior of cells by using light. This technique is very useful because it acts much faster than drugs and is also much more precise than electrical stimulation of the brain. To use an optogenetic technique, the gene for a protein which expresses a special channel is injected into a brain along with a promoter in the form of a virus. This virus then infects neurons in the brain and delivers the gene so that the cells express the protein channel. To activate the protein, a light is delivered to the neurons via fiber optic cable. The light-sensitive channel called channel rhodopsin is important in optogenetic activation. Blue light causes the channel to open, which then causes an influx of positive ions similar to that of a neuron firing an action potential. When these channels are expressed in mammalian neuronal cells, then researchers have a way to precisely control nerve activity in the brain. When a study uses channel rhodopsin in optogenetic research and neuronal activation, the figures are often displayed in blue, as seen in the primary research articles of focus later in this video. To silence neurons with light stimulation, a different channel is expressed in mammalian cells called halorhodopsin. This channel is activated with a different colored light and causes an influx of negative ions, which switches neurons off or inhibits nerve activity in the brain. To show that halorhodopsin was used in an experiment, researchers will often present their figures using a yellow color, as seen in this example. The two primary research articles of focus for the rest of this video use both behavioral and optogenetic techniques. The researchers in the Tai et al. Paper tested whether midbrain dopamine neurons could have a causal role in both induction and relief of diverse stress-induced depression-related behaviors, including reduced motivation and anhedonia. The study used a chronic mild stress protocol to induce depression in mice and then controlled depression-related states using optogenetics and assessed certain depressive phenotypes using the tail suspension and sucrose preference test. They first tested whether selective inhibition of VTA dopamine neurons could acutely induce multiple depression-like behaviors. They found that the inhibition of these VTA neurons induced a depression-like phenotype. Inhibition decreased the time spent struggling in the tail suspension task and decreased sucrose preference, which are both symptoms of depression. Figure 1c shows a baseline on and off again VTA dopamine neuron inhibition paradigm for the tail suspension task. As shown in the graph, mice in the experimental condition have a decrease in time spent struggling when the inhibition light is on. The control condition, shown as the gray line, does not change with the optogenetic inhibition like the experimental group does. Figure 1e shows similar data and results, but with the sucrose preference test. During the optogenetic inhibition of the VTA-DA neurons, the mice have a decrease in sucrose preference, as indicated by the orange experimental group. Again, the control group does not have a change in sucrose preference with the inhibition. To further test the causal link to depression-like states in the VTA-DA neurons, they also activated DA neurons to correct the depression-like phenotypes brought on by a chronic stress paradigm. They found that optogenetic activation of these neurons rescues stress-induced depressive behaviors. Photoactivation of this area decreased depressive behaviors in previously depressed mice. In the second figure, they used 4 groups for their experiment, 2 of which were not exposed to the stress paradigm and 2 that were. The gray line indicates the control group that was exposed to the depression-inducing stress paradigm but did not express the light-sensitive channel, while the blue group was exposed to the stress paradigm also, and the mice's dopamine neurons expressed the light-activated channel rhodopsin. Figure 2c shows the control group in gray, staying low in the time spent struggling on the tail suspension task, which indicates a steady depressive state. The experimental group, shown by the blue line, however, is rescued from the depressive state with activation of the neurons. This increase in time spent struggling not only increases during the activation phase, but the mice also do not go back to their depressed baseline when the activation is over. The data shown in Figure 2e has similar results but with the sucrose preference test. The gray control group stays depressed as indicative of a low sucrose preference, while the blue group shows an increase in sucrose preference during and after light stimulation. The results from this article show that bidirectional effects of the VTA dopamine neuron activity rapidly induces and enhances diverse depression-related behaviors. In another study, the researchers found that optogenetic induction of phasic firing of VTA dopamine neurons mediates susceptibility to social defeat stress. They used a sub-threshold social defeat protocol, which generated depressive symptoms in susceptible mice but not resilient mice. VTA dopamine neurons show two patterns of firing, one of which is a high-frequency rate called phasic firing. Phasic firing is well known to encode reward signals in the brain and is upregulated after repeated social defeat stress, like used in this study. Chaudhuri et al. Found that phasic stimulation of VTA-DA neurons increases depression-like phenotypes as measured by sucrose preference and social avoidance tasks. Phasic stimulation instantly induced an increase in social avoidance and a decrease in sucrose preference, which indicates an increase in depressive behaviors. Figure 2c shows the social interaction data measured 24 hours after the social defeat paradigm, which induced depression in the mice. The control is shown in white and the experimental group is shown in blue. The graph reveals that phasic activation of the experimental mouse decreased the amount of time the mouse spent interacting with another mouse, as indicative of an increase in depressive behaviors. Figure 2d shows the data from the sucrose preference test, measured over a 12-hour period. Phasic stimulation significantly decreased the amount of sucrose consumed by the mouse, which is also indicative of an increase in depressive behavior. Interestingly, these two studies show contradictory data and results in the activation of VTA dopamine neurons. The study by Tai et al. Found that activation of VTA-DA neurons decreases depressive behaviors in previously depressed or stress-induced mice. On the other hand, the Chaudhuri et al. Paper found that stimulation of VTA dopamine neurons rapidly induces depression susceptibility and depressive behaviors in mice without a repeated social defeat stressor. So, why the contradictory results? One possible explanation for these dramatic differences is that the studies used different depression models to induce depressive behavior in rodents before measuring the phenotypes. The chronic mild stressors could reduce dopamine firing, whereas the more severe stressors, like the social defeat models, could increase dopamine neuron firing. These two studies show that mesolimbic dopamine system is very complex, as different stressors can cause opposite responses from VTA neurons depending on the pre-exposure to depressive-inducing stressors and the severity of the stressor. The Tai et al. Paper used the time-intensive chronic mild stress paradigm, which is a method in which unpredictable mild stressors are delivered for months in order to induce a depressive-like state. The Chaudhuri et al. Paper, on the other hand, used the social defeat stress paradigm, which was repeated for 10 days. In this paradigm, the test mouse is placed in the home cage of a larger mouse, during which the experimental mouse is attacked by the larger mouse. Different types of stressors appear to have different effects on DA neurons, which can explain the contradictory results of these two studies. A fairly mild, inescapable stressor reduces VTA dopamine neuron activity, whereas restraint stress increases DA neuron activity. Long-term, inescapable, and milder stressors, such as the paradigm used in the Tai et al. Paper, appear to produce an adaptation of the DA system in a way that weakens its response to later stressors.
For something as complicated as the brain and as mysterious as mental illnesses, to have hopes of designing precise and specific therapies, you need understanding, you need deep understanding, and that's true no matter what the therapy looks like, whether it's a medicine or a talk therapy or a brain stimulation treatment. And what we're already seeing is that this is happening, that the insights from optogenetics have already helped guide brain stimulation therapies, whether with deep brain electrode stimulation or with external transcranial magnetic stimulation. And the possibilities, we think, will continue to grow from there, even though the main goal of optogenetics is basic science and discovery and understanding. Its influence can extend across different kinds of treatment.